Over the last several years, an HIV vaccine has moved from distant possibility to imminent reality. So we need to seriously start considering how we're going to make such a thing while maintaining effectivity and, just as important, global accessibility. It doesn't do us any good if it's too expensive, doesn't last, any other actual making sausage logistics of the thing. Now, a little bit of background. We're actually pretty decent at fighting the HIV virus. The problem is, by the time your body learns what it's looking for, it has changed shape 10, 30, 100 times. We're always playing catch up. This is compounded by the fact that the virus kills the very cells that we need to defend ourselves. It's kind of a lose-lose scenario. However, there are a few things that the virus cannot change while maintaining its dirty mission. One of those is called the envelope protein. Now, this is a structure on the outside of the virus that does the actual docking with our cells as it attacks them. That makes it the primary candidate for a vaccine target. So what you want to do is to design an envelope protein that you can inject into a patient and teach their immune system what to look for and hopefully recognize all incoming forms, not allowing the virus to really gain a foothold and do its distribution. Unfortunately, HIV has a few other tricks up its sleeve, one of which is the glycan shield. Now, this is a dense briar patch, a thick, thorny mess of sugar structures on the outside of the virus. So even if you're able to mount a defense and generate antibodies against it, they cannot penetrate and get down to the underlying protein. It makes it very difficult. For years, this has been a significant stumbling block in developing a vaccine that was effective. However, recent studies have indicated this may in fact be the Achilles heel that we've been waiting for. A number of antibodies have been found in clinical patients that specifically recognize the sugar group and can reach through the thorny bush and touch the protein underneath. That is, they require both the sugar shield and the underlying protein structure. So that's wonderful. My research involves designing and engineering cells to produce high-quality envelope protein with the correct type of sugar on them. And that's key, the correct type of sugar. It turns out most all mammal proteins have very complex, elaborate sugar groups on their proteins. The HIV sugar, by contrast, is kind of crude, if you will. So, using modern genetic engineering techniques, I've inserted a few on and off switches into cells that allow me to knock out the genes responsible for all the fancy polishing step that typically occurs in mammal proteins, or any animal protein for that matter. The reason you want to switch is that you can produce a high-quality protein, make a lot of it, and then reach in there and turn it over to another kind of sugar, making a massive batch with the two kinds of sugars typically found on HIV. So making a lot, cheaply, very streamlined, and that's what we're going to need to eradicate HIV. Thank you.